Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Amni Husna binti Abdul Aziz and I'm from Faculty Science and Technology FST, University Science Islam Malaysia USIM. This review paper is actually my undergraduate project and I have choose to review on carotenoid pigments extracted from the Rhodotorula Mucilaginosa for the food application. This is the outline for the review on carotenoid pigment of Rhodotorula mucilaginosa for the food application. Carotenoids are pigments that can be produced naturally in the cells of plant, insect, and microorganisms and are responsible for their yellow, orange, red, and purple color. They also can be produced synthetically by humans. Carotenoids are used widely in food sector as food colorant and antioxidant, either to restore the natural level of this molecule in food product or to produce fortified product. The production of carotenoid from the microbial sources are advantageous than the plant sources because the carotenoid from the plant sources are challenging to get due to the seasonal and geographic variability that us humans cannot control. So this review focuses on the potential benefit and health effect of carotenoid, particularly from the yeast Rhodotorula mucilaginosa, and the challenges and prospect of these pigments in the food industry as a food colorant. Rhodotorula mucilaginosa is an ubiquitous saprophytic yeast that can be recovered from terrestrial and aquatic environment, either the freshwater or marine. They are also commonly known as the red yeast. However, they may be appear as a colony of orange and corapine. Usually, this species is smooth, but sometimes they can be reticulate, rugose, or corrugated. The cell was oval globes with no spore formation. They also possesses asexual reproduction, and the highlighted characteristic of this species will be the production of carotenoid that can be used as natural colorant, especially in the food product. Carotenoid produced by Rhodotorula mucilaginosa are mainly torulahodine, torulin, beta-carotene, and gamma-carotene. The health benefit of the extracted carotenoid from Rhodotorula mucilaginosa are related to their antioxidative potential, protection from the sunlight or sunburn, antimicrobial potential, and also as lipid or single-cell oil producer. Rhodotorula mucilaginosa can isolate from various sources and are able to produce carotenoid as shown in the table. And previous studies have optimized the production of carotenoid by manipulating the nutritional content in the media and also by inducing mutation to produce hyperpigmented strain. The genus Rhodotorula possesses an identical or conserved carotenoid biosynthesis pathway with the production of torulahodine, torulin, beta-carotene, and gamma-carotene as the main product. Figure 1 shows the general biosynthesis pathway of carotenoid in the Rhodotorula species. The biosynthesis of carotenoid in Rhodotorula species involves several steps with the involvement of specific enzymes that catalyze each step of the biosynthesis pathway. Phytoin is the first colorless carotenoid in the pathway that been produced by the consecutive condensation of isoprenoid units. Dehydrogenation of phytoin is occurring continuously and the conjugated double bond is extended until the formation of neurosporin. Neurosporin may be transformed into lycopene or beta-carotene, and from the lycopene we will eventually get the beta-carotene, gamma-carotene, torulin, and also torulahodine. A research has been conducted by Yu et al. On antimicrobial activity of Rhodotorula mucilaginosa AY01 strain against two antibiotic-resistant bacterial strains, namely F1 and PWG, isolated from the porcine semen. They test the antimicrobial activity using the Kerbetis diffusion method. The antimicrobial activity was observed by comparing the clear zone around the carotenoid disc with the antibiotic disc. The antibiotic disc that has been used is the penicillin and erythromycin. The clear zone around the carotenoid disc were found similar to the antibiotic disc of penicillin and erythromycin. This indicates the inhibition of the bacterial growth, means that the pigment exhibit antimicrobial properties. In addition, Elsa Honte et al. Have utilizing the extracted pigment from Rhodotorula mucilaginosa in preparing the sweet candy jelly. They use yeast malt medium for the biomass growth and carotenoid production. The suitable temperature found was 28 degrees Celsius, while the pH is safe. Furthermore, the maximum growth and carotenoid production were achieved after 72 hours. It is important to evaluate the safety of the extracted pigment from Rhodotorula mucilaginosa. One important virulent factor that can produced by many pathogenic foodborne bacteria is the hemolysin. Previous study on Rhodotorula mucilaginosa has shown a strong beta hemolytic activity at 37 degrees Celsius and a weaker activity at 15 degrees Celsius. It is undeniable that in the food industry, the safety of the food product is a crucial aspect to be considered. The limitation of the review is the study on carotenoid extracted from the Rhodotorula mucilaginosa is limited by the lack of information on their biological properties. In addition, the only method to assess the antimicrobial activity of this extracted pigment is either by disc diffusion or well diffusion. Further tests should be conducted using minimum inhibition concentration (MIC) and minimum bactericidal concentration (MBC) for a more reliable results. Today, carotenoid from the yeast Rhodotorula mucilaginosa has become industrially important, especially in replacing the synthetic carotenoid in the market. They can provide the consumer with health benefit as well as helping the manufacturer in reducing operational costs because they can easily grow in a low-cost substrate. Thus, carotenoid from Rhodotorula mucilaginosa is a good candidate for commercialized and industrial use as a natural colorant for food products. As for the conclusion, Rhodotorula mucilaginosa has the potential in producing carotenoid as a promising alternative to the synthetic carotenoid in the market. However, there is less literature data found on the biological properties of this species, especially on the antimicrobial activity. Thus, more detailed study should be conducted on the carotenoid pigment of Rhodotorula mucilaginosa to discover and clarify more about their advantages and debatable information. For the acknowledgement, the authors would like to acknowledge the economic and also technical support of Faculty of Science and Technology, FST, and University Sains Islam Malaysia, USIM. Here is the list of references that I have been referred throughout the making of this slide presentation. And that will be all from me. Thank you.